את הפרס ולמשפחת חוברס על הכבוד הזה, לכבוד לי להצטרף לרשימה המכובדת הזאת. ובהזדמנות הזאת אני רוצה גם להודות לאלון, המנחה שלי בדוקטורט, על 6 שנים מרתקות ומענות. So I'll switch to English, and I'm happy to share with you today most of my findings from my PhD thesis, who focused on the central and the peripheral role of CRF receptor type 2 in mediating energy homeostasis. As you all know, a major part of the research in Alon's lab focuses on the CRF system. This is the primary mediator of the central stress response and contributes to the etiology of stress-related psychiatric illnesses, where in response to stress, the body activates a series of coordinated responses aimed to promote the survival of the organism and to restore homeostasis. The members of this family includes, of course, the CRF, which initiates the HPA axis and also acts as a neuromodulator in the brain. We have 2 G-protein coupled receptors, the CRF1 and CRF2, and other ligands are the urocortin, where urocortin 1 binds and activates both receptors, and urocortin 2 and especially urocortin 3 are selective ligands for CRF2. The general notion is that CRF1 is important for the initiation and activation of the stress response and CRF2 for the recovery process, but this is not always the case. In animal models, it is very clear that exposure to stress, whether acute or chronic stress, leads to reduced food intake and body weight loss, but in human it is much more complicated. However, it was shown that activation of both CRF receptors mediate an anorectic effect. Exposure to acute or chronic stress disturbs the behavioral and the metabolic homeostasis, and exposure to chronic stress may lead to the clinical presentation of visceral obesity, type 2 diabetes, and the metabolic syndrome. And I will focus today on the CRF receptor type 2. This receptor has 2 splice variants. The variant alpha is expressed in the central nervous system, and pharmacological studies, as well as studies that use CRF2 knockout, show that in addition to its role in mediating the stress response, it is also important in modulating metabolic rate, appetite, and feeding behavior. Actually, the knockout mouse for this mouse were generated and extensively phenotyped by Tracy Bale, who is here with us today. So here you can see the distribution of these receptors in the brain, and you can observe that within the hypothalamus, both receptors, but especially the CRF2, is expressed. The other variant, the beta variant, its major expression is in the periphery with very, very high expression levels in skeletal muscle tissue. Its ligand in the muscle tissue is urocortin 2, and it was shown that mice which lack either CRF2 or urocortin 2 have enhanced glucose tolerance. And further work done by Alon showed that CRF2 signaling functions to inhibit interaction between insulin signaling components, which leads to reduced GLUT4 translocation to the membrane. So today I will show you a work we did regarding CRF2 beta in skeletal muscle, and I will show you a bit of the approaches we took in order to study central CRF2. So in skeletal muscle, we observe only expression of CRF2 without a parallel expression of CRF1. In order to study the regulation of this receptor, we use C2C12 cells. These are myoblasts, which undergo differentiation into multinuclear myotubes upon serum removal. You can see that in the myoblast state, these cells express only CRF1 without expression of CRF2. And this profile changes throughout the differentiation process, where the CRF1 expression parallels the expression of ID2, which inhibit myogenic differentiation, and the expression of CRF2 parallels the expression of MyoD and myogenin, which are myogenic determinant factors. In order to see whether this is regulated at the promoter level, we isolated the 5' flanking region of both genes, and we subcloned them upstream to a luciferase reporter, and we followed the activation of this reporter throughout the differentiation. As you can see, C2C12 differentiation was accompanied by by inhibition of the CRF1 promoter activity and time-dependent activation of the R2 beta promoter. When we look on the 5' flanking region of the R2 beta, we can identify consensus sequence for skeletal muscle specific transcription factor. So we fragmented this promoter, so it will, each fragment will include different numbers of recognition sites, and these were also cloned upstream to luciferase, and we monitor their activation during the differentiation process. As you can see, all fragments were activated throughout the myogenic differentiation, though not as robust as the full-length promoter. But interestingly, you can see that even the shortest fragment, which is only 170 base pair long, was also activated. In this fragment, we have a recognition site for MEF2, and we mutated this site in order to see whether it is important for CRF2 transcription. And as you can see here, the mutated fragment in which the sequence of MEF2 is mutated, so it is not longer also recognized by the software that predicts consensus sequences for a transcription factor. You can see that this fragment is not activated during differentiation, and the luciferase expression is very low. So given the high expression of CRF2 in skeletal muscle tissue and its suggested role in modulating glucose uptake, we wanted to see whether it is regulated by chronic stressors. And to this end, we chose two paradigms. One is proposed as a chronic stress. This is the chronic variable stress, in which the mice undergo a few weeks of unpredictable mild stressors. And the other one is the physiological stress of exposure to high-fat diet. Interestingly, you can see that under both paradigms induced the expression of CRF2 beta in skeletal muscle. And as we know, chronic stress and obesity are hallmarks of our modern lifestyle and are correlated with insulin resistance, and this could be also due to the upregulation of the R2 beta gene. So as I've mentioned, the R2 knockout mice have enhanced insulin sensitivity. So we performed skeletal muscle gene expression array, and we found a reduction in the expression of retinol binding protein 4 in the CRF2 knockout mice, and we confirmed this by a real-time PCR. RBP4 is mainly expressed by the liver and the adipose tissue, and is known to be, its serum levels are known to be increased in insulin-resistant subjects. So when we went back to the muscles of these mice, we found that these paradigms also mediated an increase in the expression of RBP4. So to conclude this part, we saw that the CRF receptors are differentially expressed during myogenic differentiation, and we saw that this differential expression is regulated at the promoter level, and we showed the significance of the MEF2 recognition site at the 3' proximal region. We may suggest that this promoter, this very short promoter, may be further used for muscle-specific expression of target genes. In the mature mouse, we saw that different stressors modulate the expression of both CRF2 and RBP4, and this dual increase may synergistically act in an autocrine fashion to inhibit insulin signaling and therefore magnifying the metabolic complications. So clearly, CRF2 has a role in glucose homeostasis in the skeletal muscle, but in addition, the CRF system is also expressed in the pancreas. There, it was shown that urocortin 3 stimulate insulin secretion under high glucose conditions. So if we will use the developmental knockout mice, it may be hard to distinguish between peripheral or central contribution to the metabolic phenotype. So we used two approaches to target the central CRF2. The first one was to overexpress the ligand, the urocortin 3, and thus to overactivate the receptor. This we did by a combination of transgenic mice with lentiviral injections. And the other approach was to reduce the expression level of the receptor by injecting lentiviruses which express shRNA sequences. So when we overexpressed urocortin 3, we chose to manipulate an endogenous pathway in which urocortin 3 is secreted from the periphonical area, and from this site, urocortin 3 is projected to both the lateral septum and the ventromedial hypothalamus. This pathway is regulated by physiological stressors, and it involved the lateral septum, which is more related to aggressiveness and modulation of emotional processes, and also the VMH with its important role in energy balance. So what we did is we injected the mice, but we didn't induce the system yet. We didn't induce the urocotin-3 overexpression, and we checked the mice under basal state and following urocotin-3 overexpression. Here you can just see that the system works. This is slices of the lateral septums, and this is the endogenous level of urocotin-3 reaching its terminal field in the lateral septum at the basal state, and this is the amount of urocotin-3 that reaches when we induce the system. You can see that urocotin-3 pattern resembles the expression pattern of CRF2 in this site. So we tested these mice for behavioral and metabolic parameters under basal state and following overexpression of urocotin-3. This is an example for a test which measures anxiety-like behavior. In this test, the mice are placed, each one is placed in a new arena. It is called the open field test. And as you can see, under basal state, the mouse explore the whole arena. Mice likes to explore. But when we overexpress urocotin-3, you can see that the mice don't enter the arena of the center of the arena and stay close to the walls of the arena and travel a shorter distance throughout the test, and all are indices of increased anxiety-like behavior. When we tested these mice in metabolic cages, we saw that overexpression of urocotin-3, again, specifically only in this site of the peripheral area, didn't affect the mice food intake. However, overexpression of urocotin-3 did mediate an increase in the heat production of the mouse and in the respiratory exchange ratio, which is the ratio between the carbon dioxide produced to the oxygen consumed. This suggests that the mice utilize more carbohydrate and less fat as a source of energy. And it's not that the mice could choose different food source, but an interchange in fuel utilization. When we looked at the glucose homeostasis of these mice, we saw that their ability to handle a glucose load was similar when they overexpressed urocotin-3. However, when we performed the insulin tolerance test, we saw that overexpression of urocotin-3 mediated a reduction in insulin sensitivity. When we measured insulin levels, it was 20 days after prolonged exposure of urocotin-3, which I remind you is also anxiogenic for the mouse. We saw that the mice were hyperinsulinemic, meaning that exposure to urocotin-3 for 20 days at this specific site drove the mice to a pre-diabetic state. So to conclude, we saw that urocotin-3, specifically in the peripheral area, is responsible for mediating both behavioral and metabolic responses, which collectively promote coping responses to challenge. Now, to further test the role of CRF2 in the ventromedial hypothalamus in mediating or regulating energy balance, we used the complementary approach of reducing the expression level. We generated 4 lentiviruses which target the open reading frame of the CRF2 gene. We chose for the one which was most efficient for reducing the expression, and this virus or control virus were injected to the ventromedial hypothalamus of these mice, and we assessed the metabolic profile of these mice under basal state and following challenges. When we looked on these mice food intake, we saw no differences on the basal state. Actually, we found no differences in all the parameters we measured in the metabolic cages under basal state, and this further support the general or the becoming notion that this is a regulatory system and we need to challenge the system in order to see its significance. We food deprived the mice while in the metabolic cages, and we monitored their food intake in the 24 hours of the refeeding. And here you can see that the mice consumed significantly less food compared to the control groups. In fact, they ate 75% compared to the control group. And this was the exact occurrence that was reported for the developmental CRF2 knockout mice by Tracy Bell. They also consumed only 75% compared to their wild-type litter mates. There it was suggested that these mice eat less because they have an anxious phenotype. In our hands, our knockdown mice behave similarly in tests that measures anxiety, so we may suggest that this phenomena is mediated by the VMH CRF2 neurons. When we also looked on the meal pattern analysis during the basal state and the refeeding period, we saw that In contrast to the control group, the knockdown mice failed to increase the number of meal they consumed, and they also didn't prolong their meal duration. When we looked on the locomotor activity, again, we saw no basal differences. However, during the refeeding period, we can see that the control group reduced their activity level, and the knockdown mice maintained similar activity. So from the control group, we can say that the normal recovery process from food deprivation includes reduced physical activity and increased food consumption, which is very intuitive, but this is maladaptive in the CRF2 knockdown mice. So CRF2 signaling in this area is required for the adaptive recovery from food deprivation. We will go on and check whether this is related to the involvement of the CRF system in the pathophysiology of anorexia nervosa. And in anorexia nervosa, you can see high prevalence of depression and anxiety, and they are characterized by high cortisol levels. And in animal models, it was shown that administration of corticosterone or exposure to chronic variable stress, which induces depression, reduces the expression of CRF2, specifically in the ventromedial hypothalamus. In order to see which other genes may be related to this altered feeding during the refeeding period, we made some hypothalamic gene expression array. We punched the mediobasal hypothalamus, and we looked on gene expression level either under fed state or following 24 hours of food deprivation. Under fed state, we found no differences between the wild type and the knockout mice. Here you can see the differences for each genotype, the gene expression profile in the deprived state compared to the fed state, and few differences emerged, but most of them were shared by both genotypes. However, only in the knockout mice we saw a significant reduction in prodynorphin level, which is the precursor for multiple opioids which exert orexigenic effect. And only in the wild-type mice we saw an increase in the expression of the receptor for MCH, the melanocyte concentrating hormone, which is a very potent orexigenic peptide arriving from the lateral hypothalamus. So overall, in the knockout mice, we see a reduction in orexigenic input, which may explain, at least to some extent, the altered recovery from food deprivation. So to summarize, maintaining energy homeostasis in the presence of diverse stressors requires numerous adaptive responses in both central and peripheral tissues. The anatomical distribution of CRF2 and its ligands within peripheral and central tissues positioned it as key player in this field. Hypothalamic CRF2 signaling is essential for reestablishing homeostasis following exposure to challenge. We hope that these and our future data will help for better understanding of the reciprocal mechanism that mediate the relationship between stress, mood disorders, and energy balance. I would like to thank you for listening and acknowledge the people who were involved.